Hello, I am Sarah Maxwell, and I am presenting species-dependent effects of novel selective Trpm4 channel inhibitor CAB on human and guinea pig Detrusor smooth muscle excitation-contraction coupling. None of the contributors have any affiliations to disclose. This project specifically focused on transient receptor potential melastatin type 4 Trpm4 involvement in bladder excitability and contractility. Trpm4 is a calcium-activated non-selective cation channel that conducts monovalent cations. It has been identified in several cell types related to important biological functions. Previously, our lab has confirmed the presence of Trpm4 in human and rodent DSM and provided evidence that modulation of Trpm4 channels alters DSM excitability and contractility in both rodents and humans. CAB is a newer compound that has been identified as a Trpm4 inhibitor. CAB was also shown to be more selective and show a tenfold potency improvement over 9-phenanthrol, which is a commonly used Trpm4 inhibitor and only moderately selective. The objectives for this project were to provide pharmacological evidence for Trpm4 channels and the regulation of DSM excitability and contractility in human and guinea pig DSM, and to examine potential interspecies differences in the pharmacological effects of Trpm4 inhibition on human and guinea pig DSM. The methods involved isometric tension recordings of both guinea pig and human DSM, as well as perforated whole cell patch clamp recordings on freshly isolated DSM single cells. Here, we have the effects of CAB on spontaneous phasic contractions. On the left, we have traces with responses to increasing concentrations of CAB. The contraction parameter plots contain both guinea pig and human data. You can see that CAB slightly attenuated both human and guinea pig spontaneous phasic contractions in a concentration-dependent manner with maximum inhibitions of around 30 to 40% for amplitude and force. Overall, there were no differences between human and guinea pig observed for spontaneous phasic contractions. Next, we have the effects of CAB on 20 millimolar potassium chloride-induced phasic contractions. Again, traces are shown as well as cumulative concentration response plots. You can see that human tissue showed a concentration-dependent decrease. The maximum inhibition for guinea pig showed approximately 25%, while human was inhibited up to approximately 75% for amplitude and force. Last are electrical field stimulated contractions in response to increasing cumulative additions of CAB. You can see that again, human tissue showed a concentration-dependent decrease in response to CAB, while guinea pig did not. The maximum inhibition for guinea pig showed 20% for amplitude, while human was inhibited up to approximately 60% for both amplitude and force. In perforated patch clamp experiments, CAB did not change the membrane potential of human or guinea pig freshly isolated DSM cells. In human, CAB inhibited voltage step-induced cation non-potassium currents at all voltages above positive 60 millivolts in freshly isolated DSM cells for human, but it did not in guinea pig. In conclusion, CAB displayed differential effects on excitability and contractility in human and guinea pig DSM cells and tissue strips. CAB displayed inhibitory effects on stimulated DSM contractions but not spontaneous phasic contractions or cell membrane potential. From this, we can conclude that in human DSM, Trpm4 channels contribute to the regulation of DSM function under stimulated but not spontaneous conditions. Further, it is critically important to conduct translational studies directly on human DSM due to target-specific species differences, as shown here for Trpm4. We would like to acknowledge our ongoing collaboration with the urologists at both MUSC and UTHSC, as well as all funding sources for the lab. Thank you.